Hi, everybody. Thank you for joining. I see there are a lot of people still coming in from the waiting room. So welcome. Thank you for being here with us today. My name is Allison, and I'm part of the patient navigation team at the Healing Center at MGH. And today I'm so excited to be joined by our guest, Dr. James Berry from MGH, who will be walking us through our update of the platform trial, as well as some special topics on biomarkers today. So without any further ado, Dr. Berry, I will hand it over to you.
Thank you so much. Thanks for having me, Allison. And I really enjoy participating in these. I just love that we do them, and I'm always glad to be here. And so updates on the HEALEY platform trial. I mean, it's really an exciting time for the trial, and we'll see that as we kind of talk through it. Next slide. For those who don't know, I'm James Berry. I'm the regimen co-lead for Regimen C. I'm at Mass General Hospital, and I'm an ALS clinician and researcher, and I do spend a fair amount of my time thinking about biomarkers and endpoints and how we can apply those to clinical trials. And so we'll talk a little bit about that later. Next slide. So again, I think, maybe many people are familiar with the platform trial idea. Maybe some are not as familiar. And so just to explain it, in a typical clinical trial, we're testing one drug and often testing that against placebo. And oftentimes it's, half of the people receive placebo and half receive the active study drug. And at the end, we have an answer for that one particular drug. And then we kind of take the whole thing apart and start over for another trial, almost like making a movie. You have to get all of the parts of that together, and then when you're done, and you go on to the next one. And the HEALEY platform trial is really a new way to approach that. And the idea is to bring efficiency to this. We know that developing drugs for ALS is difficult, and we know that the faster we can go, the more drugs we can test. And the really wonderful news is that we have more and more really viable, really promising ideas for what should go into clinical trials. And so we have this demand. And this new platform trial approach allows us to test more drugs more efficiently. And there are a few reasons for that. But one is that we use a similar kind of a protocol for each of them, which cuts down on design time and startup time. We also have the same, what we call vendors, which is, the central lab and the central pharmacy and medical monitoring and all these behind the scenes pieces that we have to put together contracts for, and all of that takes time. That's all in place. And then another thing about this is that we have all the sites lined up, and the sites are enthusiastic about being trial centers for this. And I think that's really important for keeping momentum and also speed. It allows us to start up each of the new trial regimens, each new trial within the trial. We call them trial regimens, and start those up faster. And then there's the idea of a shared placebo. So here you can see regimen A, B, C, D, and E, each in a different color. A is zilucoplan, B verdiperstat, C is CNM-Au8, D is prodopidine, and E is trehalose. And each of these is randomized 3-to-1. Now, from a statistical perspective, we do better. We have more power to detect an effect of a drug if it really is effective, the closer we get to sort of a 1-to-1, a 50% on placebo, 50% on drug. But at the same time, we wanted to think of a way to do that differently so that we were reducing the placebo burden. And the way that we do that in this trial is by sharing the placebo across these different regimens. And that's one of the real benefits of this kind of trial. Each regimen has a randomized portion and then an open label extension so that everybody who participates has a choice of then participating further and knowing that they're receiving the study drug. 2022 is a remarkable year. We will be getting the results for the first 4 regimens, A, B, C, and D. And E is the only one that is currently enrolling, and we're looking forward to adding to that as well. Next slide. So anytime we do a trial, we need to have some measuring stick to say, is the drug working? Is it slowing the disease in some way? And the primary endpoint for this trial, as it has been for so many of our trials, is the ALS Functional Rating Scale Revised, or ALS-FRSR, which, as many of us know anyway, is a 12-item questionnaire, and it asks about activities of sort of function throughout the day. And it's been used in many, many trials, and it is an imperfect instrument, but it is one that we know a lot about, and we can do a lot of pre-planning for these kinds of trials. The secondary endpoints are change in respiratory function, and we use the slow vital capacity to measure that. Change in muscle strength, and we use handheld dynamometry to measure that, which is essentially, how hard can you push against a meter that's held in somebody's hand. And then that's a quantitative number that we can use. We also look at survival, and then what we call treatment-specific biomarkers, and I'll talk a little bit about those. We have a number of exploratory endpoints, and we're always looking at safety. The drugs that are coming into this trial, tend to have at least some data in humans that would guide us to think that they're safe, and that if there are things that we need to monitor, we know how to do that as well. This is not the sort of very first early stage trial. Next slide. So I want to talk a little bit about these exploratory endpoints and how we're developing them, and how we see them contributing to this trial, and how we think building on our knowledge of them will contribute to better, faster, more efficient trials in the future. So I'll just start at the top. DNA, or genes, are the building block for our cells. There are lots and lots of things that we can look at in the DNA. Really, the lowest hanging fruit is to look at ALS causative genes and understand whether people with a certain gene mutation, and about 15% of ALS is caused by a gene mutation, and the rest is not caused by a mutation in any single gene. There may still be a genetic component, but it might be more complex. But we know about this, these 24, more than that, causative genes for ALS, and we can measure those, and then we can look at whether people with those gene mutations are balanced between the two arms in the trial, between placebo and study drug, and also, is there a subgroup that responds some different way to the drug that we're testing? And there are some examples of that in trials that we've done. The example we think about is one of lithium, and a small subgroup of people who had a mutation in a certain gene seemed to behave differently. And so there's now a trial going on in Europe looking at just that subgroup and to see whether they have a beneficial response to lithium. Beyond just those ALS causative genes, there's a whole developing field of pharmacogenetics. That is, how do our bodies process drugs? It may be that some of us process drugs much more quickly. We metabolize them and get rid of them more quickly. And so the levels in some people may be lower than the levels in other people. And we can measure that in blood, but we can also use DNA potentially to understand those effects better. And we may find genes that influence how rapidly the disease progresses, or what somebody's risk factor is. And those may have a role in understanding trial results. And so we are collecting blood for DNA, and we're doing genome sequencing so that we can begin to ask some of those questions, which is different than previous trials. Neurofilament is a biomarker that we can measure in spinal fluid or in blood, I should say, and in blood. So we can measure it perfectly well in blood. Neurofilaments are the scaffolding proteins for neurons, and they keep those neurons, they sort of build the neurons themselves. And we know that in people with ALS, neurofilaments are spilled at higher levels into the blood. So everybody has some neurofilament in their blood and their spinal fluid. It's a low level, and it just has to do with, we think, regular turnover of neurons that happens throughout our lifetime. But when there's more stress on the neurons in a disease like ALS, particularly, we see an elevation in neurofilaments in the blood and spinal fluid, and that tends to be fairly stable over time. Now, just to be very clear, this is not specific to ALS. Any injury to neurons will cause the neurofilament to go up. So it turns out in ALS, there's more of an elevation than in many diseases, but it's not specific to ALS. But we may be able to follow neurofilaments, and we think that higher levels of neurofilaments may be detrimental or may reflect a faster disease progression. And if a drug slows disease progression, we may see a decrease in neurofilaments. And so we can look at them that way throughout this trial. We also think that at baseline, we can categorize people into sort of higher and lower neurofilament levels, and that may be beneficial. And we can also put this into our statistical models as a way of really kind of helping us hone down on what is the effect of the drug. And that's a complex process, but one that essentially says, if we take neurofilament, the variation in neurofilament out of the picture, if we correct for that, then what's the effect of the drug? And being able to do that with something like neurofilaments is really beneficial. So that's something that we're actively doing as well. We also have the opportunity to look in blood and urine and CSF for other biomarkers. So one candidate biomarker that we'll look at is urinary P75. This hasn't been tested nearly as much as neurofilaments, but P75 is a protein, a signaling protein that seems to go up as the disease progresses. And we may be able to follow that in urine. It'll tell us about, we hope that it'll tell us about the disease, and it may even tell us about an effective treatment. So we're excited to begin to kind of develop that. The other thing I would say is that our biomarker work is really turning corners in understanding inflammatory biomarkers, things like chitinases, which we can measure in blood and in CSF. And we have the opportunity to explore those and even use some very broad testing techniques like proteomics, metabolomics, which means looking at all the proteins in blood, or all the or all the small molecules, metabolites, we call them, in blood, for example. And by getting a landscape of all of those markers, we may be able to then use really sophisticated algorithms. We might call them a form of artificial intelligence or AI analysis to really understand which ones are important in the disease and potentially in response to a drug. So there's a lot of opportunity in biomarkers in what we call biofluids, blood, urine, CSF. And so that's a really exciting thing that we're collecting, we're storing. We have very strict protocols for how we collect and process and store these. And I think we'll learn things about the trials and also for years to come about the disease from that. So really exciting. And we're doing a speech analysis. So anybody who's participating in the trial will know about the speech analysis. We're having people make recordings of things that they read, but also recordings of sort of syllables that are alternating. And we can use this to follow in a very quantitative way the progression of the effects of the disease on speech. And it's really one of the first times that we're putting this into a trial setting. We've done a lot of analysis of this over time in people who are not receiving a specific treatment. And we really see that we can have a very quantitative measure that does change over time and could be really valuable in assessing whether a drug is having an effect. This is certainly the largest time that we're putting this into an ALS trial. We have used it to good effect, actually, in looking at the effect of Nuedexta in the past. And now we're applying this to many regimens at the same time. So exciting to see what those results will show us in conjunction with the primary results. And then finally, home spirometry. And some of this was motivated by the COVID pandemic, putting this into the trial. But it's also something that we've been looking for ways to get more data about home spirometry for some time. And this seemed like a great time to do it. So we'll have a lot of data about how we compare data that we're getting at home, and people in the trial are putting a lot of work into this compared to data that we're getting in the trial at the centers. All of these things are really building a whole new way of looking at data coming out of a trial. And we think ultimately changing paradigm for how we do these trials. So this is a really exciting thing Our goal is to do more and more efficient trials, to have better ways to quantitate the disease, faster ways to get to the answer of whether a drug is working. And this is how we do it. And to be able to do it in a trial setting is really exciting. Happy to answer questions about this as well, but we do think a lot about this. Next slide. So just as far as progress, as of today, we've had 131 people sign informed consent. This is specifically talking about regimen E individuals that would be relevant to regimen E. 88 of those individuals have already been randomized within regimen E. These are people who are really devoting, a huge amount of time, effort, and making this trial go. And many of you are on the phone, both in regimen E and in other regimens in the trial. And this really is a partnership, and we have recognized that from long before we started this trial, and certainly from the start of this trial. This is a community effort. We are doing this together as partners, and we appreciate everything that people in the trial are putting into this to make it a success. Much of our motivation for doing things that can be done at home spirometry, home recordings of voice, is in service of trying to make trials like these easier over time so that more people can participate, and people who are participating can do that in as easy a way as possible. We also want to get to answers faster, as I know all of you do. So thank you to everybody who's a part of this, everybody who's screened for the trial, whether you've been able to participate or not, people who have just spread the word about the trial. Any part that anybody has had in this is incredibly valuable. I think that may be the end of our slides. Next, let's see if we have one more. Next slide. Oh yeah, we do. We have two more. So just for people who may not be participating, we have 47 sites that are currently actively recruiting for regimen E. They're listed here, and certainly you can use the QR code by pointing your camera on your smartphone at it, and it will take you to this map so that you can explore this more. And next slide. That doesn't work. Then we also have contact information here for Allison, who's on the phone, who did the intro today, as well as Catherine. And they are our patient navigators and really are a central resource for people living with ALS who might be interested in this trial or want to understand it more. There's a phone number here, 833-HALT-ALS, and the email healeyalsplatform@mgh.harvard.edu. You can sign up for the ALS link using the QR code on the right, and then there's the weekly webinar registration using the QR code on the left. So we do have an update next week, and we have Tafiq coming in to speak, who's the site investigator at UPMC in Pennsylvania on September 15th. And then Mariah Connolly and Megan Hall, who are site monitors for the platform trial, are coming on September 22nd. And I'll just put in a plug for them. I think this is a role that many participants and people who hear about trials don't know about. The monitors are the ones who come to each of our sites as we run these trials and really look at all of our books, all of the papers that we write on, all of those papers that people see shuffled around during our visits, and make sure that we're recording information correctly and uniformly, and doing that in a way that will be acceptable when the regulators want to look at this and we have a positive trial. It's remarkably important for trials like this. We are managing millions, tens of millions of data points, and sometimes in trials like this. And they all have to be right, and they all have to be thought out. And so that is a role that really, really makes the trial tick. So interesting to hear their perspective. I know we have some questions, and I think Allison, you were going to help moderate.
Yeah, thank you, Dr. Gray, for that rundown. I think we can stop sharing the slides now. We had quite a few questions that came in around the subject of trial results. Can you talk a little bit about where we are in understanding the data on the first 4 regimens?
Yeah, so we're expecting to have these results, really in early autumn of this year. And we're looking, so regimen D started enrolling a little bit behind A, B, and C. So A, B, and C started enrolling around the same time. And so regimen D may be slightly behind A, B, and C, but we're looking forward to those in early autumn of this year. Really an exciting time for the trial.
Yeah, definitely. And Dr. Berg, can you explain a little bit further, kind of set the expectations? I know we're all talking about, oh, we're going to have results, we're going to have top line data. What is it that we should actually expect, and how does that relate to actually getting access to these treatments, should they be positive?
Yeah, it's a really good question, and one that I think is worth pausing to talk about a little bit. So first of all, oftentimes we'll see that the first results to come out are what we call top line results. And the top line results are really the ones that we put up on that slide in the middle somewhere that say what's our primary endpoint and what are our key secondary endpoints. And those are really the things that we've designed the trial to read out most critically. And in an overall sense, so for all the participants, not subgroups, not necessarily, more nuanced kind of evaluations. That's our top line result. The programming for all of that is done, sort of along the way as we run the trial. And those are the first things to come out, along with, I mentioned at the time, but along with safety data. And that safety data is really key for understanding whether development will move forward, or what the prospects of an approval would look like. And so because that data is most straightforward and mostly how we've designed the trial, that's the first data to come off the presses. And that is often the first thing that you'll see. So when we see a press release, not just about this trial, but about any trial, the first press release to come through is often one that says, here are the top line results, and follow-up results will come. Sometimes you may see a press release that has the top line results and some more nuanced results as well. And that's because sometimes the top line results can't really be interpreted without some of the kind of supporting more exploratory analyses or subgroup analyses. And so sometimes we'll see those things come out at the same time. And there'll often be some mention of safety that was found to be acceptably tolerable, or no major safety signal. A more nuanced safety analysis often follows that. All of the exploratory analyses follow that. Many times a subgroup analysis follows that. And by follow, it's hard to put an exact timeline on how long until we get some of these other results, but they can often take weeks, many weeks to really become clear, because once we get out of those top line results, then there's a lot more nuanced analyses that we have to work through. And then finally, there are analyses that we'll sometimes hear called post hoc analyses. These are things that were not written into the protocol, that were not a part of our initial plan, but allow us to explore questions that come up as after the results come out. We say, oh, well, that's interesting. We see this trend. Maybe we should be asking this other thing as well. And we have a nuanced relationship with post hoc analyses. They can be very important. They often answer critical questions that come up as we look at the results. They are also, it's sort of like calling your pocket in billiards. It's a more impressive shot if you make it when you call the pocket. Because it may just be chance that you get that ball in some other pocket. And that's kind of how we think about post hoc analyses too, that, they can be incredibly informative and they're really important, but we also do need to be cautious in how much weight we put on them. And so I think that drives a lot of the discussion.
Okay, thank you for taking the time to explain that further. Question for you: can a person living with ALS have neurofilament level measured in a commercial lab currently?
The answer to that is no, because Previously, the answer was no. It remained really in the experimental realm until very recently. There are not many labs that run this, but there are a couple of labs that will run this now. And so we can run this in clinic. I think there's an even more nuanced answer to that, which is we have a lot of experience with neurofilament now in observational studies. We're beginning to have experience with it in clinical trials. We do not have much experience in using it in the clinical realm. And the difference here is that when we think about observational studies that tell us about the disease and clinical trials, we're thinking in groups. How do groups of people respond? When we're thinking on a clinical level, we're thinking about individuals. And those two uses can be quite different. And so I think we need to really, we're going to have a time of getting our legs under us to understand what these results mean clinically and how do we use them clinically. Are they a part of the diagnostic process? Well, some people with ALS have an elevated neurofilament and other people don't. That can say something about progression, but it's not the only thing that tells us about progression rates in the future. So will we use it in diagnosis? Will we use it in prognosis? What do we do about the fact that it doesn't always match what we end up seeing? It's not a perfect predictor. Those things are things that we're going to have to work out on the clinical realm, and that's important to know as a person living with ALS who's speaking with your clinician about whether you send that test and how to use it.
Perfect. So regarding the biomarkers that you went through with us, can you clarify, have you identified new biomarkers, or are these the biomarkers that have been being looked at for ALS?
Yeah, so we're always, it's sort of, I think this is kind of an ongoing process. So there are biomarkers at different stages of understanding. Neurofilament really leads the pack in ALS for a biofluid biomarker of the disease. And, you know, again, as I just said, we don't understand everything about it. We're just beginning to see how it behaves in clinical trials. And we're just beginning to see how it works on a clinical level, on an individual level. But we really have the most evidence about that one. There are other ones that we've looked at a lot. I mentioned urinary P75. Well, P75 is a protein that is expressed on neurons when they're developing, so in infancy, and also when they become ill. And it gets cleaved off and it shows up in our urine shortly after that. And so, you know, that's one that seems to increase over time in people with ALS, but there have only been a couple of studies that have investigated that. And the number of people who are having that done is increasing because there are now a number of other studies that are looking at it. So we'll learn more about how reliable that is and over what time course does it change as we have more samples that are tested. There's another category of protein called chitinases. And there have been a few studies looking at chitinases, but it's a category of proteins. So there are actually a number of proteins we have to look at and see how they behave. And so we've had a few studies looking at those, and I think we're beginning to see bigger studies looking at those. I think there is an elevation in chitinases in people with ALS, but you know, exactly what that tells us, you know, does it correlate with future progression? Does it tell us something about the pathology? Does it subgroup people? We still have a lot of work to do there. So you can see just from those examples that there are different stages of development, but we're working on all of these in parallel. And then there are a host of biomarkers that we're just beginning to ask these questions about. And, you know, that it's an ongoing process.
That's for sure. Okay, a somewhat specific question, I'm just hoping that you can answer. There's on the call, there's a participant in the EAP for CNM-AU8 and is also taking riluzole and radicava and wants to know the timing. Can you please clarify whether they can take the 3 meds simultaneously in the morning?
Yeah, so I'm going to step away from answering that. What I would say is the most important thing is that you stay in touch with your study center and the docs on your trial. And so I don't want to step in the way of that. What I will say is that there are studies that we sometimes do to look at whether two medications can be taken at the same time, or whether they can go through the same IV, or whether they can go through the G-tube simultaneously or one right after another. And if we haven't done those studies, sometimes we have to tell people to separate them, either in how they take them by mouth or through a G-tube or through an IV, until those studies are done. So what I would say is just important to keep in touch with your center. And as with any question that comes up like this, it's a quick question to the center, and hopefully they'll get right back to you with an answer.
Very good answer. One more specific question that you may be able to answer, Dr. Berry. The AMX 0035 EAP, is there a different dosage for slow progressors versus fast progressors?
So the AMX, so stepping away from the platform trial for a moment.
Yes.
So the AMX 0035 has one dose that really was the target dose in the Centaur trial, and is the target dose in the Phoenix trial. And that is essentially one packet in the morning and one packet at night. So one packet twice a day. And it can be taken at a lower dosage than that. So, you know, if people have, in those trial settings, if people have some reason to go down, it can be taken once a day instead. To my knowledge, it's not stratified by sort of fast or slow progressors, but rather, you know, especially in the EAP, the discussion between the treating physician and
we're going to take, I know we're running out of time here, but I do want to take one more question that we had come in. We have a participant who travels from Hawaii to Dallas to participate in our trial, and wants to know if there is a way to attend appointments in person on the mainland instead and participate virtually for the EAP program. Do we have virtual options for our EAP programs?
So yeah, so for every trial or EAP program, I mean, first of all, let's take a step back and say this has really changed a lot because of the COVID pandemic, and actually pushed a lot of our ideas forward dramatically. So we've had some of these ideas, and began implementing them, but we were pushed to do this with the pandemic. And so now, when we're writing protocols, we really sort of think about whether visits have to happen in person or whether they can happen remotely. And I think we're really trying to sort of make as many things remote as possible, but we do also have to keep an eye on safety. And so different protocols will have a different approach to what can be done in person and what can be done virtually. That's driven a lot by the safety aspect of it, or the potential safety aspect of it. You know, what do we need to monitor? When do we need to see people and do an EKG or do blood draws or have eyes on people? And that'll vary a lot by protocol, and it might be different for the same drug. It could potentially be slightly different for an expanded access protocol and a trial protocol.
Perfect. Okay. I do want to mention, in case people didn't notice, that there was a question that came in where people could view the webinar schedule and who will be attending as presenters and such, and that is on our website. I dropped the link into the chat for you, but it is on the Mass General website. And before we leave, Dr. Berry, is there anything else that you would like to share?
Just that I want to thank you for having me. I just really value being included in these, and I've done a number of these webinars, and I always enjoy it, and I just love the community that we have, and I want to thank everybody here for being on and taking your time.
Yes, and thank you so much for joining us today, and for everybody who joined us. I know we did have a few questions about non-HEALEY platform trials, and usually we do try to answer those if there's time. Next week, Drs. Paganoni and Sokovich will be with us, and hopefully after our usual update, we'll have time to take some of those questions. So I encourage you to come back and ask them again. Hope everybody has a great night, and we will see you next week. Thanks so much.
Thanks a lot. Take care.
Bye-bye.